Hi everyone, welcome back to Neurobiology at Providence College. I'm Joe DeGeorgis. Today we're going to talk about neuronal electrical action potentials in the work of J.Z. Young and Hodgkin and Huxley. Let's first talk a little bit about the anatomy of a nerve cell. A nerve cell has, of course, a cell body, and the cell body contains the nucleus here, which is where the DNA is located, of course. And it has dendrites. There are these branches that emerge from the cell body, and they receive signal from either upstream neurons or from specialized sensory cells that sense the outside world, like touch, smell, taste, and so on. Then it has an axon, this portion here, and then it has the terminal end of the cell where the axon splits, it bifurcates and forms two branches, and then those go on to form other branches that connect through synapses to either downstream neurons or to muscle at the neuromuscular junction. So these are dendrites that receive signal from other upstream cells, the axon, which propagates the electrical action potential, and then the terminal end of the cell here where it synapses to other downstream neurons or, as I mentioned, muscle in terms of the neuromuscular junction. How neurons conduct electricity and propagate electrical action potential came from the discovery by J.Z. Young of a very thick nerve cell that exists within squid. This is now referred to the squid giant axon. For this lecture, I asked you to watch a few YouTube videos showing fishermen collecting squid off of Plymouth, England for Young and other scientists. This collecting process also happens off of Cape Cod through the Marine Biological Laboratory in Woods Hole using the fishing vessel Gemma. I was a crew member on this boat in my early 20s. I became a diver for the laboratory, and now I'm a summer researcher. J.Z. Young is a zoologist, and he was taking a look at squid. And the squid body is cone-shaped, like an ice cream cone. And then it has a head, and these are cephalopods, which means they have their feet attached to their heads, like cuttlefish, octopus, and nautilus, along with the squid. And they have a big eye, and they have a fin on each side of its body. And it turns out that the ice cream cone part, the mantle of the squid, is hollow, and water can enter into this cavity. Now there are organs in here. There are gills, and there are two brachial hearts, and a systemic heart, and digestive tract, and all of those sorts of things inside the body. But water can enter and exit this cavity. And squid use this as a form of locomotion. So they expand through expanding this muscle. They draw water into the mantle, and then they contract this part of the body, and it shoots the water out in this direction, and then the squid moves in this direction. It's sort of a jet propulsion mechanism. So there are a number of lobes that make up the brain, and they're behind the eye here, and there are neurons that run from the brain of the organism down to a stellate ganglia, that is a synaptic connection and a region that communicates with a large number of neurons that innervate the mantle. So there is one giant axon that innervates this part of the mantle, and there's a second and a third and a fourth and so on. And I'll get to that in a moment. You can dissect the animal by cutting along the edge with a pair of scissors. Just put the scissors in here like this and cut along the edge. And you can open the mantle up so that it has this triangular shape. So you're cutting the ice cream cone and then flattening the cone out. And there, as I said, there are two gills in here, and there's a couple of brachial hearts, and there's a systemic heart, and other organs that are found inside the mantle. But these are very easy to remove. And inside the mantle, as I said, there are two neurons that come down from the brain, and they connect to the stellate ganglia. And from the ganglia, there are a number of axons that innervate the muscle of the organism, like this. So these are axons irradiating away from the stellate ganglia where the cell body is. And these all conduct electricity, ultimately, or we know that now. Like this. And these axons bifurcate, that is, they split, and then they split and innervate the muscle. So this is a connection, a synaptic connection between the neuron and the muscle at the neuromuscular junction, which we'll talk about later in the course. But at any rate, these are like roots of the axon, and they feed out into the muscle. So if we go back to our simplified diagram of the anatomy of a neuron, again, we said this is the axon. This is the terminal end of the cell. This is the bifurcation and multiple sub-bifurcations. And the dendrites that we said receive signal from other upstream neurons or specialized sensory cells. And the structure that we want to talk about for the sake of this conversation is the axon, which is here beyond the cell body and before the first bifurcation of the axon. So this part here, which is sort of the trunk of the tree, is the axon. And interestingly enough, in squid, this axon is about 1 millimeter in diameter, which is really large, 1 millimeter. Ours are about 1 micrometer in our brain, which is about the thickness of a human hair. So 1/1000th the size of the squid axon. The large size of the squid giant axon provided Hodgkin and Huxley with a unique opportunity to be able to put an electrode inside the squid giant axon and one outside of the axon and measure the potential across this membrane. And in general, the difference between the inside and the outside is about minus 70 millivolts, so that the inside is more negative relative to the outside at rest. They were also able to stimulate the neuron and trigger an electrical action potential and found the potential to look something like this. So if we redraw our axon in this direction with our electrode here and one electrode here, we can measure the potential across the membrane, and the potential is minus 70 millivolts, and the inside of the axon is more negative than the outside of the axon. So Hodgkin and Huxley and others were able to do a very large number of experiments to tease out the details of how the resting potential is obtained and how the action potential is propagated. First of all, the inside of the cell contains a large amount of protein, and proteins in general are negatively charged. Of course, there are amino acids that have positive charges on them, but overall proteins are negatively charged. Along with proteins, there are very high concentrations of potassium ions inside the cell and high concentrations of sodium ions outside of the cell. So sodium is high in concentration outside of the cell and low on the inside of the cell. And potassium ion concentrations are high inside the cell and low outside of the cell. And it turns out that the action potential is propagated by channels in the membrane that selectively allow either potassium or sodium to pass through the membrane. So you could have a potassium channel like this, K+, and you could have a sodium channel, Na+. And if the channel opened, then the ion would flow down its concentration gradient. That is, potassium, in this case, would go from the inside of the cell to the outside of the cell. And if a sodium channel opened, sodium would run down its concentration gradient from the outside of the cell to the inside of the cell. And we'll talk about that in a little more detail in a minute. Now, it turns out that normally when there's no action potential and we are at rest, at the resting potential, the potassium and sodium channels are closed. But it turns out that the potassium channels are leaky. So some potassium leaks out through the channel and allows some potassium to flow down its concentration gradient. But the proteins, which are negatively charged, also attract the potassium ions. So the negative charge of the proteins attracts the positive charge of the potassium and holds some of the potassium inside the cell. Finally, we have in the membrane sodium and potassium pumps, and these pump 3 sodium from the inside of the cell to the outside of the cell, 3 sodium ions, and it pumps 2 potassium ions into the cell through the hydrolysis of 1 ATP molecule, and it gets hydrolyzed to a DP diphosphate from triphosphate plus a free phosphate. So it uses the energy in breaking that bond to move the pump and pump 3 sodium ions out and 2 potassium ions into the cell. So, to say that one more time, to summarize that, at rest, when the cell is not propagating an electrical action potential, there's a high concentration of protein inside the cell that holds on, that attracts positive potassium ions. Some of the potassium can be transported out of the cell through leaky potassium channels. The potassium is running down its concentration gradient from the inside of the cell to the outside of the cell. There's a high concentration of sodium outside of the cell, and we have sodium-potassium pumps that pump sodium out of the cell and potassium into the cell at the expense of the hydrolysis of 1 ATP molecule. And as this goes on, the axon reaches an equilibrium, and the equilibrium is such that the inside of the cell to the outside of the cell has a resting potential of minus 70 millivolts. Okay, let's go back to the anatomy of a nerve cell. So if we have a neuron with its cell body here with the nucleus, we have the axon, which propagates the electrical action potential, this portion here we said, and then the terminal end of the cell, which bifurcates and synapses to other downstream cells, and finally dendrites that connect to upstream cells, like this. If we take a look at the axon, let me draw it a little bit bigger, like this. we said that at rest it is at minus 70 millivolts. So if we draw a graph, we have a resting potential here of minus 70 millivolts. Like this. And we said that's a result of a lot of activity that's going on inside the cell, and that there's very high potassium inside the cell, for instance, and high sodium outside of the cell. We have proteins with negative charges inside the cell that attract the potassium ions. We have potassium channels that are leaky so that some of the potassium can leave the cell, run down its concentration gradient. And then we have sodium and potassium pumps that are pumping 3 sodium ions out of the cell while it pumps in 2 potassium ions and hydrolyzes ATP to ADP and phosphate, like this. So if we measure the potential across the membrane, we would get a reading of minus 70 millivolts. Now it turns out that other neurons upstream connect to the dendrites through a synaptic connection. So, the terminal here of the upstream neuron, that's synonymous with this here, is synapsing with the dendrite. And if this neuron up here is firing for whatever reason, then it releases a neurotransmitter that travels through the synaptic cleft, and it binds to receptors in the cell membrane of the dendrite, which we're going to talk about in more detail. But if that's the case, this could be a neurotransmitter gated or a ligand gated ion channel that allows sodium to enter into the cell. And if you have sodium entering the cell at this point, but it flows throughout the cell, then sodium is increasing in the cell, and that causes the potential to increase because you have more positive sodium ions entering the cell. I mean, the sodium is positively charged, a positively charged ion, and if it enters the cell, then the resting potential is going to go from minus 70 millivolts in a positive direction towards 0 millivolts. So you're going to get an increase in the resting potential. Now, if the signal is not that strong, that is, you don't have very much sodium entering the cell, then the sodium dissipates and the resting potential goes right back down to 70 millivolts. If, however, you have enough sodium that you reach the threshold at something like minus 55 millivolts, then you hit a point in which there's an all or none response, in which the sodium channels open. And sodium now rushes down its concentration gradient from the outside of the cell to the inside of the cell, and the potential continues to increase beyond 0 and then reaches its peak. At that point, the sodium and potassium channels close. The sodium channels close, and the potassium channels, which are slower to react, open, and potassium rushes out of the cell. So because sodium was rushing into the cell, positive ions were coming in, and the potential was going from minus 70 millivolts past the positive point to something like 40 millivolts plus 40 millivolts. And then the sodium channels close and the potassium channels open. So sodium close, potassium channels open. And then as the potassium channels, the potassium, which is also a positive ion, rush out of the potassium channels out to the outside of the cell, the potential starts to decrease and goes below zero, below the threshold, then below the resting potential, and then the potassium channels close. So it actually goes below the threshold because the potassium channels are slow not only to open, but slow to close. So this is potassium channels closing. And this dip below the resting potential is called the overshoot. And then once the potassium channels close, the cell goes back to its resting potential at minus 70 millivolts. And of course, at rest, the sodium and potassium pump continues to pump, pumping 3 sodiums out and 2 potassium ions in. We have negatively charged proteins that are attracting positively charged potassium ions. We have high sodium again on the outside of the cell, and we have leaky potassium channels that allow some of the potassium to run down its concentration gradient back to a resting potential of minus 70 millivolts. Okay, that's it for today. See you guys next time.